Dear students, Assalamu alaikum to all of you. Among one of the important biomolecules, we are having nucleic acids. Nucleic acids are macromolecules of utmost importance. They are polymeric compounds of nucleotides and are called as polynucleotides. A nucleotide is formed of three components: heterocyclic nitrogen base, second, the pentose sugar, and the third one, a phosphate group. Formation of nucleotides takes place whenever these three substances combine: heterocyclic nitrogen base, pentose sugar, and phosphate group. Heterocyclic nitrogen bases are of various types: adenine, guanine, thymine, cytosine, and uracil. A polynucleotide chain is formed by interlinking of large number of nucleotides by 5-3- phosphodiester bonds. These bonds link the 5- carbon of a pentose of a nucleotide with 3- carbon of pentose of adjoining nucleotide. The bond between sugar and phosphate nucleotide is called an ester bond, while the linking of phosphate group to two sugar nucleotide is called phosphodiester bond. So the backbone of a nucleic acid chain is formed of alternatively arranged phosphate and pentose sugar molecules, while the nitrogen base stick out inward from the pentose sugars of the backbone. Nucleic acids are generally of two types: deoxyribose nucleic acid and ribose nucleic acid. Deoxyribose nucleic acid, what we call as DNA, it occurs in the nucleus of all living cells except some viruses, for example, tobacco mosaic virus. It was later reported in a number of cell organelles like mitochondria, plastids. That is why we call these cellular organelles as autonomous. Watson and Crick was the first group of scientists that gave the double helix model of DNA. Later on, Michelson and Stahl in 1958 gave the confirmation of this double helix model of DNA. This model of DNA was proposed on the basis of biochemical research that the DNA is a double helix that is made up of two polynucleotide chains. The double helix is wound in such a manner that there is a torsion, just like as we find the torsion in the different microthreads of a rope. It has been found whenever we see this double helix, the diameter of the double helix is 20 angstroms, and there is a difference of 3.4 angstroms between each nucleotide. There are two types of grooves, major grooves and shallow grooves, present in the DNA. The DNA in the nucleus forms genes. A gene is a definite segment of DNA, which is chemically called a cistron. A cistron determines a character. Whenever there is duplication in the DNA, sometimes there are some mutations. That mutations lead to the alteration of the structure of the DNA. That is why these genes get mutated. Ultimately, they prove disastrous for the generation of characters or traits. DNA forms the genome. DNA combines with the nucleotides to form nucleoproteins. These nucleoproteins are designed in the form of small dot-like structures. They are called as nucleosomes. The nucleosomes are the backbone of the chromosomes. Chromosomes are present in the nucleus, made up of nucleoproteins that has a fixed number in a particular cell. For example, in human beings, there are 23 pairs of chromosomes or 46 number of chromosomes. DNA undergoes duplication. DNA undergoes transcription, in which DNA synthesizes the RNA, and DNA also undergoes a very important function that is called a translation, in which DNA synthesizes proteins which determine the character. That is all for today.